everyone, today we're going to be talking about cellular respiration. We'll start off with an introduction, we'll look at aerobic respiration, anaerobic respiration, as well as fermentation. Energy is necessary for all types of cellular activity, such as moving organelles around the cell or creating different biomolecules. All reactions involved in the capture, storage, and use of energy within a cell are collectively known as metabolism. The two most important metabolic pathways on Earth are photosynthesis and cellular respiration. During photosynthesis, carbon dioxide and water are converted into organic molecules such as glucose, releasing the waste product oxygen. This reaction is anabolic and requires an input of energy, and that energy comes from sunlight. During cellular respiration, organic molecules such as glucose are broken down, releasing energy and forming carbon dioxide and water as the waste products. The breakdown of organic molecules is exergonic, which means that the energy of the reactants is greater than the energy of the products, and so the reaction releases energy. There are three pathways that organisms use to obtain energy from organic molecules: fermentation, aerobic respiration, and anaerobic respiration. Fermentation is the least efficient of all three processes because it's only a partial breakdown of the sugars. Aerobic respiration and anaerobic respiration are similar to one another, but aerobic respiration requires oxygen to be present while anaerobic respiration does not. Aerobic respiration is by far the most efficient out of all three processes. Aerobic respiration is often referred to as simply cellular respiration. During aerobic respiration, organic molecules will be broken down, releasing energy in the form of ATP. Aerobic respiration requires the presence of oxygen, as oxygen is the final electron acceptor in the electron transport chain. Anaerobic respiration is similar to aerobic respiration, but the main difference is that the final electron acceptor is something other than oxygen. For example, organisms can use molecules such as carbon dioxide or nitrate. The energy stored in organic molecules is released through the transfer of electrons, and this released energy is ultimately used to make ATP. Oxidation reduction reactions involve the transfer of energy through the transfer of electrons. Oxidation refers to the loss of electrons, and it is normally accompanied by the loss of a hydrogen atom. Reduction is the gain of electrons, and this usually involves the gain of a hydrogen atom. Oxidation and reduction reactions always occur together. The oxidizing agent would be the agent that is doing the oxidizing, so it itself would be reduced and would be gaining electrons. The reducing agent is the one that does the reducing, so it itself is going to be oxidized and it's going to lose electrons. Oxygen is a really good oxidizing agent because of its high electronegativity. It has a very strong pull on electrons. During aerobic respiration, electrons are passed from organic molecules to electron carriers, namely NAD+ and FAD, onto the electron transport chain, and then finally onto the final electron acceptor, which is oxygen. If we have a look at the overall reaction of aerobic respiration, we see that glucose becomes oxidized, losing electrons to form carbon dioxide. Oxygen, on the other hand, is reduced, so it gains electrons and forms water. Aerobic respiration takes place in three main stages: glycolysis, the Krebs cycle, and then the electron transport chain. Glycolysis takes place in the cytoplasm of the cell and results in the production of 2 ATP per glucose molecule. The Krebs cycle, on the other hand, takes place in the mitochondrial matrix. During the Krebs cycle, another 2 ATP are produced. The final stage is the electron transport chain, and this occurs in the inner mitochondrial membrane. That membrane is highly folded to fit as many electron transport chains in as possible. And the reason for this is because the electron transport chain is the stage that produces the most amount of ATP, producing about 32 ATP per original glucose molecule. Let's have a look at the overview of aerobic respiration. So we have glycolysis occurring in the cytoplasm. During glycolysis, glucose is going to be broken down, forming pyruvate. When this happens, electrons are going to be transferred onto NAD+, and NAD+ will be able to carry those electrons to the electron transport chain. Glycolysis is also going to produce 2 ATP. Pyruvate will then move into the inner mitochondrial matrix and enter the Krebs cycle. During the Krebs cycle, it's going to be broken down, releasing electrons and releasing carbon dioxide. Those electrons will move on to electron carriers, once again NAD plus and FAD, which will carry those electrons to the final stage of aerobic respiration, the electron transport chain. During the Krebs cycle, once again, we produce 2 ATP per original glucose molecule. The electron transport chain that is occurring in this inner mitochondrial membrane, what we have is a series of proteins embedded in that inner mitochondrial membrane, and they're going to pass electrons from one protein to the next until they finally end up on that final electron acceptor. This process is going to generate 32 ATP. We need an input of oxygen because it is the final electron acceptor, and oxygen is going to be reduced, so it's going to gain electrons and produce the waste product water. Let's start off by looking at glycolysis. If we look at the name glycolysis, glyco is referring to the sugar and lysis means to split. So we're literally splitting that glucose molecule in half. Glucose is a 6-carbon compound. So each of these gray balls is representing a carbon atom that we find inside the glucose molecule. And during glycolysis, we have a number of enzyme-regulated reactions that are going to break that glucose down into 2 pyruvate molecules, each having 3 carbon atoms. This process is going to produce 2 ATP, and it's also going to generate our electron carrier NADH. We'll get 2 NADH per original glucose molecule. The first step in glycolysis is the conversion of glucose to fructose 1,6-diphosphate, and this step requires an input of 2 ATP. So these phosphate groups that get attached onto the glucose molecule originate from these ATP molecules. After the formation of fructose 1,6-diphosphate, this molecule will be cleaved, forming glyceraldehyde 3-phosphate. So we'll produce 2 glyceraldehyde 3-phosphates. Glyceraldehyde 3-phosphate will then be converted to pyruvate. During these steps, NAD+ will pick up electrons to form NADH, which is an electron carrier. NADH will then transport those electrons to the electron transport chain. The formation of pyruvate from glyceraldehyde 3-phosphate also results in the production of 4 ATP. Since we use 2 ATP in the first stages of glycolysis, the net production of ATP from glycolysis is 2 ATP per glucose molecule. Between glycolysis and the Krebs cycle, we have an intermediate step. During this step, pyruvate is going to enter the mitochondrial matrix via active transport. Pyruvate is then going to be converted to acetyl coenzyme A. This conversion occurs through the loss of a carbon atom in the form of carbon dioxide. We also form more NADH, and that NADH will move on to the electron transport chain. Coenzyme A then drops off acetyl at the Krebs cycle. The 2 carbons from acetyl join to a 4-carbon compound known as oxaloacetate. This forms a 6-carbon compound called citric acid. Citric acid is then oxidized back to oxaloacetate through a number of enzyme-regulated reactions. During this process, citric acid is going to lose carbon in the form of carbon dioxide. We're also going to form electron carriers, NADH and FADH2. For every acetyl coenzyme A entering the Krebs cycle, 1 ATP is produced. This means that the Krebs cycle produces 2 ATP per original glucose molecule. Let's move on to the final stage of aerobic respiration, the electron transport chain. The electron transport chain takes place in the highly folded inner mitochondrial membrane. So if we're having a look at this diagram, we have our outer mitochondrial membrane, we have the inner mitochondrial membrane, and then we have the mitochondrial matrix. This is where the Krebs cycle has just taken place. On the other side of that outer mitochondrial membrane, this is where we find the cytoplasm. Embedded in that inner mitochondrial membrane, we have a series of proteins. And these proteins are going to transfer electrons from one protein to the next using redox reactions until those electrons ultimately end up on our final electron acceptor, which is oxygen. So firstly, we need to get electrons to this electron transport chain. Those electrons are going to come from electron donors. Our electron donors, in the case of aerobic respiration, are NADH and FADH2. NADH was generated in glycolysis as well as the Krebs cycle. FADH2 was generated in the Krebs cycle. Let's just use NADH in this example. NADH is going to be oxidized to NAD+, losing electrons to this first protein in the electron transport chain. Electrons are then going to be moved from protein to protein via redox reactions until ultimately they end up on oxygen, our final electron acceptor, and water is formed. So oxygen is our final electron acceptor and it is reduced to water. The energy released due to the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. This is active transport. The hydrogen ions are moving against their concentration gradient from an area where there's a low concentration to an area where there's a high concentration of hydrogen ions within that intermembrane space. This maintains a hydrogen ion concentration gradient with a great amount of hydrogen ions within this intermembrane space. These hydrogen ions would like to move from this intermembrane space back to the mitochondrial matrix along their concentration gradient. But they cannot simply diffuse across the lipid bilayer because of these fatty acid tails sequestered inside the lipid bilayer, which are hydrophobic. The hydrogen ions therefore move back into the mitochondrial matrix through a special protein known as ATP synthase. The potential energy from the hydrogen ion concentration gradient is then converted into chemical energy in the form of ATP. As the hydrogen ions flow through ATP synthase, the energy released is used to attach a phosphate group to ADP and form ATP. Some environments lack oxygen, such as swamps or the intestine of animals, but we still find microorganisms living there. How are these microorganisms producing energy? Well, they'll either be using anaerobic respiration or fermentation, two processes which can occur in the absence of oxygen. An obligate aerobe is an organism that has to have oxygen present in order for aerobic respiration to occur. An obligate anaerobe is an organism that has to live in an environment that lacks oxygen. We also have a facultative aerobe and anaerobe. A facultative aerobe is an organism that lives in an environment with oxygen but can also survive without oxygen. A facultative anaerobe, on the other hand, is an organism that lives in an environment without oxygen but can still survive in an environment if oxygen is present. Anaerobic respiration is very similar to aerobic respiration. There will still be glycolysis, there will still be the Krebs cycle, and there will still be the electron transport chain. The difference between aerobic and anaerobic respiration is the final electron acceptor. In aerobic respiration, that final electron acceptor is oxygen, but in anaerobic respiration, the final electron acceptor is something other than oxygen, such as carbon dioxide. The amount of ATP produced during anaerobic respiration depends on the final electron acceptor that's used, and it will always be lower than aerobic respiration. The reason for this is because oxygen is such a strong oxidizing agent because of its high electronegativity. So it pulls the electrons very quickly through the electron transport chain and therefore generates more ATP. Organisms that use anaerobic respiration include bacteria and archaea. I'm sure that most of you have heard that cows have a bad rep of creating methane gas, but in fact it's not the poor cow's fault. It's actually the bacteria living inside its rumen. Inside the rumen there's no oxygen, and so the bacteria are performing anaerobic respiration using carbon dioxide as the final electron acceptor. When carbon dioxide accepts those electrons, it is reduced to methane. Fermentation is the partial breakdown of sugars in the absence of oxygen. During fermentation, the only stage that occurs is glycolysis. There is no Krebs cycle and there is no electron transport chain. So you can see that fermentation is going to be way less efficient than aerobic or anaerobic respiration because per glucose molecule we can only make 2 ATP. Now remember, during glycolysis, NAD+ accepts electrons and forms NADH. So in order for glycolysis to continue, NAD+ has to be regenerated. So glycolysis will be followed by post-glycolytic reactions that regenerate the NAD+. These reactions do not produce any more ATP. Their sole purpose is just to deal with the pyruvate formed and regenerate NAD+, so glycolysis can continue. There are two types of fermentation pathways depending on these post-glycolytic reactions, the lactic acid pathway and the alcohol fermentation pathway. During lactic acid fermentation, glycolysis occurs, so glucose is converted to pyruvate. In this process, we make 2 ATP per glucose molecule, and we also generate NADH. The pyruvate is then going to accept electrons from NADH, and thus regenerates the NAD+ and forms lactate. Many bacteria are able to perform lactic acid fermentation, and we use them to produce things like yogurt and sauerkraut. Our muscle cells are also able to perform lactic acid fermentation. Let's say that you go for a really strenuous run. Your muscle cells are going to require a large amount of ATP to keep up with that activity level. Aerobic respiration on its own is just not going to be enough to provide the amount of ATP that your muscle cells are demanding, and so the muscle cells turn to lactic acid fermentation. The buildup of lactic acid in your cells is what irritates the nerve endings and leaves you feeling all sore and painful the next day. This is why after strenuous exercise, it's always a good idea to exercise lightly the next day. Light exercise will get the blood flowing, will clear out that lactic acid from the cells. And in fact, the lactic acid will be taken to the liver, where it will be converted back to pyruvate, and then that pyruvate can enter the Krebs cycle and continue down the pathway of aerobic respiration. During ethanol fermentation, glycolysis will occur as normal, and what will happen to the pyruvate is it will first lose carbon dioxide and form acetaldehyde. That acetaldehyde is now going to accept electrons from NADH to regenerate the NAD+. And when it does that, it forms ethanol. Yeast and some bacteria are able to perform alcoholic fermentation, and so we use these organisms to produce yummy things like bread, as well as fermented beverages such as wine, beer, and cider. Alright, so this diagram is a comparison of all three pathways. We can see that all three pathways include glycolysis, and in fermentation, that's it. It stops at glycolysis. We make 2 ATP per glucose molecule, and we form pyruvate. That pyruvate is further broken down in post-glycolytic reactions so that our NADH is regenerated to NAD+, and glycolysis can continue. In aerobic and anaerobic respiration, however, the pyruvate is going to be converted to acetyl coenzyme A, which will enter the Krebs cycle inside that mitochondrial matrix. The Krebs cycle is going to generate further electron carriers, NADH, FADH2, and those are going to move on to the electron transport chain, along with NADH coming from glycolysis. In aerobic respiration, our final electron acceptor is going to be oxygen, whilst in anaerobic respiration, it'll be some kind of molecule other than oxygen, which won't pull the electrons as strongly, so the amount of ATP we produce in this anaerobic respiration will be less than aerobic respiration. However, the least productive out of all three would be fermentation. So we've come to the end of our session. Please go ahead, pause your video, answer these review questions by yourself, and then come back and we can go through the answers together. Firstly, what process do fermentation and aerobic respiration have in common? Well, that's glycolysis. What is the final electron acceptor of aerobic respiration? It is oxygen. And in fermentation, well, we have two different types. In our lactic acid fermentation, remember that the pyruvate itself is the final electron acceptor, accepting electrons from NADH. In alcohol fermentation, the final electron acceptor is acetaldehyde. So pyruvate is first broken down to acetaldehyde, and then that is the molecule that accepts electrons from NADH, forming ethanol and regenerating the NAD+. How much ATP is produced in aerobic respiration and fermentation? Aerobic respiration is 36 per glucose molecule. Fermentation is 2 per glucose molecule. Most carbon dioxide from cellular respiration is released from which process? And that would be the Krebs cycle. So when that citric acid is broken back down to oxaloacetate, the carbon that is lost in the form of carbon dioxide. What is the purpose of post-glycolytic reactions? Well, they're to get rid of the pyruvate so it doesn't build up and become toxic in the cells, but also to regenerate that NAD+ so that glycolysis can continue. What is the main objective of cellular respiration? To make ATP. And then the last question, where do the stages of aerobic respiration occur in the cell? We have glycolysis occurring in the cytoplasm, the Krebs cycles in the mitochondrial matrix, and then the electron transport chain is in the inner mitochondrial membrane. I hope you've enjoyed this session with me today and that you've learned a lot, and I look forward to seeing you again next time.